Hello everyone, this is Priyanshu Choubey, Assistant Professor of Zoology and University. Today we will talk about soil biology and biomass. Soil organisms, they are beneficial and harmful mechanism. Soil biology and biomass. Uh, Soil biology, interaction and function, types of interaction in an ecosystem. The living organism interact with each other for their survival. The types of interaction among the living organism are mutualism, corporations, commercialism, and the emilism participates, and exploitation, the inter-specific competition, uh, antagonism, and the predictions. Microbe and microbial interaction. Before we will discuss about the soil biology. Soil biology is the study of a microbial and the fungal activity and the ecology in soil. This organism include earthworm, nematodes, protozoa, fungi, bacteria, and different arthropods. When these bacteria, fungi, and the animals die, the living organism it in the soil, it the dead creating the organic matter. These reactions are depend on the soil, types of soil, and on the decision that humans make. The biology of a soil also considers how biotic interaction in the soil influence the in the important soil process of decomposition and nutrient recycling. Soil organism, a single of a gram of soil can contain a billion of microorganisms. Soil organisms contribute to great biodiversity in soil, and the interaction with each other, plants and animals in the soil to provides us with ecosystem functions that are essential for a life. Some important rules includes nutrient cycling or decomposition. Classification of soil organisms, soil fauna, earthworms, protozoa, nematodes, termites, springnels, etc. These are a fauna. Soil flora, bacteria, algae, fungi, etc. These are a soil flora. Microbe and microbe interaction, interspecific interaction among microorganisms may be positive or negative, either for one partner or both. Microbe interaction carry out many ecological significant soil process. For example, lichness or such as the important biological interaction between a fungi and more cyanobacteria, green algae. Green algae, which types of relationship among organisms and their interactions. So, play a dominant role in a decomposition of rocks in a soil development process. The partner cyanobacteria provide a fixed nitrogen and a photosynthesis to the symbiotic partner fungi, and exchange cyanobacteria get a mineral nutrient from the fungi in a natural In a nature members of a bacteria nature members of a bacteria actinomycetes and fungi coexist exit they carry out decomposition of organic residues in the group efforts. Plant microbes interaction, the plant micro interaction may be a beneficial and detrimental depending on the types of partner. The association, like a legumes, rhizobia, beneficial plant, endophytes partner, mycorrhiza, azora, anabena, sugarcane, Gluconidobacter and rice, Pontia rice, Azerosperium, etc. are some of the prominent example of a beneficial plant microbial interaction. The word mycorrhiza derived from a Greek word myco means fungus and rhiza means a root. The mycorrhiza implies the association between a non-pathogenic fungi with its host roots. There are three major types: mycorrhiza association, androphic, and orchidaceans, and the non-shitting and avascular type, and actinomycorrhiza or the shitting types, and actinomycorrhiza or the arceus types. Avascular mycorrhiza, also called AM fungi, they establish a symbiosis with 70% of the vascular and the non-vascular plant families. The edges and sedges and the cruciferia, some chiropodaceae, sugar beets, and a certain quite are the expected expectation. The mycorrhiza groups provide a distinct morphological structure within a root tissue like vessels and the avascular. The The accepted fungal hypha penetrate the epidermal cells, inter cortex, and the grow intercellularly. The intercellular using a hypha coils. Finally, they penetrate the cortical cells and form a vesicle where the most nutrient and the water exchange occurs. Mycorrhiza phase of collector nutrient NPK, zinc, etc., and the water form the reason which are the not accessible to plant root and deliver uh, them in the cortex cell. In the exchange of fungal hypha get the photosynthesis from the host root. The mycorrhiza fungi also play the significant role in the soil aggregation. The mycorrhiza fungi also play a significant role in the soil aggregation and the orbitode, monotropic, and the actinomycorrhiza association. The fungus forms of a hypha metal over the root surface, and the hypha penetrate the root and the proliferated intercellular offer from the heritage. The group of mycorrhiza form the symbiosis with the gymnosperm and the Angiosperm species, and this group of the fungi is a taxonomically diverse. The approximate 6,000 species. The EMA fungi are able to assess the organic form of the nitrogen and phosphorus and have a, spirophytic ability. Abrotite and the monotropes mycorrhiza transport the calcium and the nitrogen phosphorus to their host. Likewise, Ericord mycorrhiza primarily increase not only host access to nitrogen but also to phosphorus and calcium and iron. Based on the size, the microorganism is greater than 2 mm in the width, and mesoorganism 0.222 mm in width, and microorganism less than 2.0 mm in width. Ecological functions have behaviors that subsist on a living plants, directivators and the subsist on the dead plants, debris. Uh, Predator that consume animal and fungivores that eat a fungi and bacteria that eat a bacteria. Parasites that live off but do not consume other organisms. Decomposer decompose the organic matter. Soil content and the anamorphous number of the living organisms that cup of the under distributed. The native soil may contain like organism and number of bacteria 2000 200 billion and protozoa 20 million and fungi 1 lakh and nematodes 1 lakh and arthropods 50 number. The soil organism bacteria single called organism that are the microscopic ranging in the shape from a sphere roots to spermites li- eight legged animal too small to see with the unaided eye the feet on a smaller organism and soil organism matter. Earthworm create a horizontal and the vertical burrows in the soil, allowing the H2O to enter the soil and the carbon dioxide to leave the soil. Capable of a decomposition that organic matter and the releasing the energy in the time in them so that other organism can use it. Mix soil layers together to mix the horizons layer distinct at the same time they are adding organic matter to the different layers. Actinomycete treated like a bacteria that often resemble fungi and they are not as great as the uh, abalance as other types of bacteria. They have a vital role in the soil. They help decompose the organic matter into a humus and the release nutrient in the process of doing so. So capable of the producing antibiotics and the aid in the fighting roots disease and responsible for producing a that sweet and tree smell of soil. Algae present in the many soil provided that they are in the moisture and the sunlight available. The most commonly types and found in the soil are the blue-green algae and the green algae. Plant an important role in the maintaining the soil fertility, especially in a tropical soil. And the organic matter is matter to the soils when they died also help prevent in the soil erosion. Nematodes, tiny, segmented worm-like organism that mostly live on other organism or soil organic matter. Protozoa, single-celled organism larger than a bacteria that moves in a water films and the fed on a bacteria on a soil organic matter. Rotifers are microscopic animals found in the mainly freshwater environment and it in moss soil that move by a swimming and the crumbling. The spring tells six lignant animal that often have a tail-like structure folded beneath the body that can be used for jumping when the animals is threaded. Some bigger soil organisms include mice, moles, and ground sparrows, etc. They mixed the soil through their burrowing activity and contribute to the formation of a horizon. When of a soil organism, there are three main roles that soil organisms perform in a soil: decompose organic residues, recycle nutrient form organic residues, enhance soil structure. Soil organisms clear away the degrade organic debris such as the dead plants, animal, and the dung, and use it as the source of the food and a nutrient for a plant. Soil organisms are also responsible for soil physical fertility as well. They help the formation of a good soil structure, nitrogen fixation. Soil organisms live in the mutually beneficial relationship with plants, enhance the plant's nutrient by increasing the nutrient nitrogen uptake from the atmosphere rhizobium, or a phosphorus uptake from the soil by a mycorrhiza. Soil organisms play a key role in the nutrient cycling, as soil organisms decompose a complex material, or as they consume other organism nutrient are the converted from to another. They allows the nutrient to be made available for us by other plants and the soil organism. They decompose the organic matter, which acts as a storehouse for energy and nutrient for a plant and other nutrient organism use. We to encourage a beneficial soil organism, add organic matter to the soil. So, soil organism requires a food source from soil abundance, a compost crop residues, and other or mulch. Use organic mulch. It stabilizes soil moisture and the temperature and adds the organic matter. Water if effectively soil organism require and the environment that is dump like wrong outer sponge and but not soggy between a 50 Fahrenheit to 90 Fahrenheit. Harmful effect of a soil organism: certain micro fauna with the rodents and the moles and the termites may greatly damage a crop. Most plants roots are the infested with the nematodes, so seriously the successful growth of certain crops becomes difficult and expensive. Most of the plant diseases are caused by the microflora like bacteria, fungi, and actinomycetes. Food-related alliance with will occur. Agriculture productivity can decrease. Fauna microbial interaction: the soil fauna may establish uh, legume rhizobia, establish symbiosis by a formation of root nodules where the prokaryotic partner fix atmospheric dinitrogen into a nitrate with the help of the nitrogenous enzyme. The rhizosphere is the interface the plant roots, the soil where the intense microbial and the fungal activity occurs. The term rhizosphere was coined, coined by a German scientist Hiltner 1904 and defined as a zone of soils under the influence of a living roots. Usually, rhizosphere extend about 2 mm out of from the root surface. The root epidermis cortex is zone cloined by the microbes is termed as the endorhizophyum. The zone of the co- colonization outside the root surface is termed the actinorhizosphere. Ectorhizosphere, the zone where the endorhizosphere and the ectorhizosphere merge in the probably, where some of the most intimate plant and the microbial association occurs in termed as a rhizoplane. The rhizosphere zone usually harbours a higher microbial population than the bulk soil. The mechanism of the plant microbes, the interaction in the rhizosphere zone or the oh, rhizo deposition by the living plant's roots. The rhizodeposits attract the certain member of the native soil microbes, especially the uh, copy- copytrophs who adopt the resources-rich habit. Several members of the microorganisms solubles the mineral nutrient like phosphorus, zinc, etc. form their insoluble fractions, making them available for a plant uptake. It's partially a competition among microbes and the colonizing rhizosphere. Competition between roots and microbes for oxygen, even a nutrient under limiting and condition, and the production of the growth hormones by a rhizobacteria. Funnel microbes interactions while Fina may establish mutual elastic or the actinostic association. Microorganism, the sleeping beauty hypothesis, illustrate the mutualistic relationship of the earth worm with their gut microbiota. Certain members of the soil bacteria dwell in the gut of earthworms, and upon indications of organic distress by the earthworm, the gut microbiota get activated and the proliferated, and and the create the digestive system. As lizard earthworm find it easy to digest the integrated material. In contrast, Mia funnel members also feed and the resources with the high bacteria colonization and the fungi. Funnel and funnel interaction. A wide range of the soil. The fauna are living together. The soil habits where they compete with each other. The very pieces of the fallen leaves, dead animals, tissues, and even predation. Among the living members, the predatory nematodes feed on a protozoa, and the non-predatory arthropods or the similar nematodes. Many predatory arthropods feed small groups of the arthropoda or a small invertebrate. Beetles and ants feed a predator nematodes and the micro and the meso arthropods. Even many bigger earthworm species feed on a small ones. Typical periparatous relationship of earthworm and the hammer-headed worms in a dispersed. Such interactions such are ecological important in maintaining population balance among a funnel members and transfer of energy for through a tropical leaves. Soil enzymes enzymes play an important role in the cycling of nutrient in nature, and because they are the activity and sensitivity to agriculture practices, they can be used as an index of the soil microbial activity and fertility. Biological indicators such as the soil microbial biomass and enzymatic activity seems to be better indicators since they respond much more quickly to both natural and orthopathogenic factors in comparison with the other variable. Origin and classifications. So, the total biochemical activity in a soil compressed of the series of reactions crystallized by the enzymes. The enzymes may be broadly categorized into intercellular and extracellular enzymes. Enzymes located within a living or dead organism are the intercellular, and those are enzymes which are the distinct from their points of the origin or termed extracellular enzymes. Extracellular enzymes can be free or a bond to soil colloids. The accumulated extracellular enzymes may be bound to a microbial cellular components. For example, in non in non-proliferating cells, intact dead cells and the cellular fragments are not associated with the cellular components originating there from a microorganism and the soil fauna and from plant roots. Accumulated extracellular enzyme mostly located in the liquid phase in the soil solution or bound to soil components. In the contrast, intracellular enzymes for proliferating microorganism, plant roots, and the soil fauna are located in the living organism and active roots. The soil enzymes can be classified as oxy, oxy, oxyreduction, transferase, and hydrolysis, and the lysis. Some of them are constitutive, which are the produce a constant amount without the regards to the physiological demands and the concentration of the substrate, while other are the introducible produce expressed only under the condition in which the clean adaptive value. Enzyme activity. Enzyme activity, dehydrogenase, so activity of dehydrated DHA reflected the total range of oxidative activity. Soil microorganisms may be considered as a good indicator of the uh, oxidative metabolism in the soil, thus of a microbial activity. Dehydrogenase oxidize soil organic matter by transferring protons and the electron from organic substrate to inorganic acceptors. Urease enzyme is responsible for the hydrolysis urea fertilizer. Nitrate formed to carbon dioxide with nitrate and then carbon dioxide with the contaminate rise in the soil pH and nitrogen loss to atmosphere through nitrate volatilization. Urease can be produced by a bacteria, yeast, and fungi and algae as well as the plants. Cellulase in the group are hydrolytic enzymes that catalyze the breakdown of beta 14 linkage bonds in a cellulose. Complete degradation of the cellulose requires at the least three enzymes and beta 14 glucolase, which attacks the cellulose chains of at a random exobeta 14 uh, gluconase, which remove a glucose and celluloses from the non-reducing end of the cellulose chains, and the D-glucosides plays a key role in the breakdown of a lower molecular weight uh, dicarbohydrates, such as the hydrolysis of the cellulose and the other water soluble cellulose. Duxness to a glucose, the activity of a glucosidase is a particularly stable and has a lower seasonal variability and is a very sensitive to soil pH and the soil management practices. Therefore, the can be used as a good biochemical indicator for measuring ecological changes that result from various agriculture management practices. Uh, phos- Phosphates, the enzyme catalyze the hydrolysis of the organic phosphorus compound and transfer into the inorganic form of the phosphorus, which is then assimilated by the plants and microorganisms. Agriculture soil contain phosphophosphates in a varying amount and depending on the microbial count. The amount of organic materials, mineral and organic fertilizer, tillage, and the other agriculture practices. Phosphates are typically adaptive enzymes, and their activity increase when the plant available phosphorus content decrease. Aerosulfates. The enzyme is involved in the hydrolysis and the aromatic sulfate esters into the phenol and the sulfur by a splitting on oxygen sulfur linkage, a mineralization and ester sulfate in a soil. Mm. Sulfides has been directed in a plant, animal, and microorganism and soil, and is in only one of the many types of sulfate is involved in the mineralization of the ester sulfur compound. Earlier sulfates is a mostly adaptive enzymes, and the synthesis of by a microorganism may be controlled by the carbon and sulfur content in the soil. Proteins proteins carry out of protein hydrolysis in a soil. Proteins are the unique uniques on the soil. These originate from microorganism, bacteria, actinomycetes, and fungi, plants, and the animal. Extreme many of these enzymes are the extracellular as a large number of activity. Proteins are not large to be absorbed by a living cells. Protein add to the soil readily decomposed by the mm, protease and the peptides into a smaller membrane permeable peptide and amino acid. The latter are further metabolized and with the release of ammonia and amylase together with the cellulose and the inverts from the group enzymes that are the responsible for the rate and the course of the decomposition of the plant materials in the soil. Amylase in the group of starch and hydrogenic enzymes, in the which the most important is alpha amylase, which is convert starch into the glucose, and the other oligosaccharides, and the beta amylase, which convert starch into the maltose, and the alpha amylase are the synthesized by the plant and animals and the microorganism organism, while a B amylase is produced mainly by a plant. Factor affecting a soil enzyme activity. Numerous factors can influence enzyme activity in a soil. Natural parameters, the seasonal change and geographical location, in situ distribution, and physical chemical properties, content of organic matter, clay or a biomass, and the orthoponic management environment, population Additive such as a fertilizer, pesticides, salts, heavy metals factors may influence the production activity, catalytic behavior, and the presence of the soil enzymes. This seasonal variability is manifested on as the combined the influence of the soil temperature and moisture and vegetation cover, which is the directly and indirectly influence the soil enzyme activity. For example, the activity of inverts and the amylase in the supper Legion under the hard beech forest and beech forest is the least in the summer and the greatest in the winter. The dehydrogenous activity of forest stand and the different stage of regeneration and altitude is a high in a May. At the low altitude, the highest phosphate activity is observed in the month of May, whereas in the higher altitude, activity are highest during March and September. The activity of the soil enzymes may differ according to the geographic location. The highest
Surface
enzyme
which
activity
breaks into pads of a
usually
different size and
observed in a
and shape.
temperate
Shape
environment.
and size can also be observed directly from
In
the cleavage
the
of the pads in the
Arctic
soil
tundra,
profile.
uh,
Indirect method involves a
community is
measurement of the
characterized by the very
size
low temperature
distribution
and the
of stable
activity of the
aggregates,
enzymes such as the
stability
urease,
of aggregates of the
phosphates,
soil
and the sulfates are the
property,
significant in the soil.
which is the function of soil structure. Soil is
Denitrification
indirect
enzymes
method, size distribution of
are
stable aggregates.
active in the
So,
desert
dry or
soil,
wet sheaving techniques are used for
demonstrating
determination of
tolerance
different
of the
size
extended
stable
period of
uh,
disease
aggregates. Indirect
dissociation in
dry
situ
sheaving
in
techniques,
situ
the
distribution of the soil
proportion
enzymes
of
activity
a
decrease
stable
with the increasing depth
aggregates
and increase with the
against
increasing organic
vibrating
matter
action,
content water being the
stimulating
major
of the
cellular components of the soil. Microorganisms in the
scoring
soil help
effect of
in
wind is
the
determined which the
catalytic process,
provides and the
especially in the
index for a
hydrolytic
characterizing
reactions.
the
Salt influence the catalytic
susceptibility
behavior
of
of
soil to
enzymes
wind
by
erosion.
action
In
of their
wet
activity and affecting the
shaving
solubility of the
technique, size
enzymatic
distribution of
protein
water
through
stable aggregates in determined
salting out effect.
measurement of the stability
The
of aggregates.
furthermore, both the
It is
factor may
evaluated by
have
the degree of the
impact
which soil
on the
aggregate
osmotic potential
resist
of the soil.
dispersion.
Either
Several
increase in
indicates
the
have
soil
been
concentration of the
developed, such
decrease
as
in
the
the soil
stability
water
index,
content may
structure
lead to
coefficient, and dispersion
hyper,
coefficient. Stability
hypotic
index
osmotic
and
pressure, result
is
in the decreased
the
metabolic
difference
activity
between
of
a
the soil
percentage silt and clay as
microbiota, or
determined by
in
the
the
mechanical analysis and
osmotic
that the obtained
uh,
by suspension of
desiccations of the
soil sample
microbial
in
cells.
water.
Soil pH
The
effect,
great the
availability
difference,
of
the better in
nutrients
the
control
soil
and decomposition and diversity
structure.
of the microbial
Structure
community alter
coefficient is given by
and the
uh, D minus
equilibrium
S upon
soil
S,
phase and imparts the
where
plant
D is a
response.
percentage
If
of particles
it affects
less than
the activity and the
0.25 mm
enzymes due
in
to a pH
diameter as
sensitivity
determined by the mechanical
sensitivity
analysis,
of
and
a
the S
amino
is the
acid functional
percentage of
group
the
that
aggregate
the
is
alter
smaller than 0.025 mm in diameter as
conformational
determined by
and the
the wet
chemical
sieving
change
method.
of
High the value of the
ammonia
structural
acid
coefficient,
essential for
the better is
binding
the
and
soil
the
structure.
catalyze. The
Measurement
pH
of
can
the
also
soil
affect the
properties, bulk
enzyme activity
density,
by influencing the concentration
infiltration
of the
rate,
inhibitors and
hydraulic
activities in
conductivity,
the soil
aeration,
solution and the effective
available soil
concentration
water,
of the
degree of
substrate. The
compaction
optimum range of pH is
of soil
close
may be
neutrally.
used for evolution of soil
The
structure.
optimum
So
pH range of
indicates
various
of a
enzyme
soil
like
structure amount of a different size soil aggregates is represented by the single value of the
glucose
comparing the
oxidase
structural
optimum
states
range is
status of
6.0,
different soil
urease
or the same soil
9.0,
under different management
protest 3.5
particles
to
among a
5.5,
various indicates
alkaline
mean
phosphate
a weight
phosphate
diameter of the
9.0 to
aggregates
10.9. So in a natural soil, microbes communities are
is
affected
a
by
commonly used
the soil
index of
texture.
soil
Soil
structure.
texture affect the relative use of amino acids and
The
carboxylic acid and
mean weight or diameter
polymerase
of
present in
aggregates
the soil.
gives an
Microbial
estimates
populations with
of
which
a weighted a
decades
percentage
enzyme
of the
activity. Soil
average
enzyme
size of
activity
all
can be
the
affected by the
aggregates. The
presence of the
proportion by
nature of
weight
plant
of
cover.
the given size
Although most
fraction
of the
of the
soil
aggregates
enzymes
to the total
activity
sample weight is a
originate from the
multiplied by the
microorganism
mean
plant.
Diameter
Roots are
of the that
and the
frictions.
important
The
source
sum
of
of
the
these products for the
extracellular
all
enzymes
size
in a soil.
frictions
The
gives a
enzyme
mean
activity
weight
tends to be
diameter
a
of
higher
aggregates
close
in a
to the root
mm.
surface in all the
Uh,
of the crops species. Plant roots stimulate enzyme activity by creating advantageous condition of the microbial activity. It crop system that
management of a
involves
soil
both
structure. So
leguminous
the
and the
general practices
non-leguminous
for the
plants.
improvement of the maintenance of
The
the soil
leguminous
structure tillage
rhizosphere shows
operation be
a
carried out
higher
within the
activity
range
of the
of the
acid
optimum
nutrient
moisture
and the
condition to
like
ensure least
alkaline phosphates
destruction
as
of the soil
compared with the
aggregate. Zero and minimum tillage
non-leguminous plant
practices be
rhizosphere.
adopted to
Long-term
maintain
leguminous
adequate
cover cropping
aeration
and the
and the
direct
reduce the loss of organic matter
incorporation of
through
a green manure
oxidation
increase
of
the soil enzyme
erosion. Soil
activity
surface
due
be
to
kept covered with the crop residues to
uh,
protect
the
aggregates
enhancement
from
of
the
a
beating
soil
action
organic matter
of
and the
raindrops.
stimulation
Check
of the soil microbial activity.
wheat
The
emergence
influence of the
and
inorganic
add organic
fertilization on the
matter, incorporation
soil
of the
enzymes
crop
activity
residues and
depends
the
on the dose of
manures into the soil for the
um,
stabilizing
it
aggregates.
depends on the dose of the fertilizer
Introduction
and the time of
of
its
legumes
application, the types
in
of
crop
climatic
rotation
condition
coupled
and the
with
enzyme
the
itself.
application
Nitrogen
of the
fertilization
phosphatic
can increase
fertilizer.
or
Green
decrease the
manuring
activity
and
of some
cover
carbon,
crops
nitrogen,
are a good
phosphorus.
source of organic matter.
Uh, Cycling
Integrated
enzymes like
use of
cellulose,
organics
urease,
and
and
fertilizer for crop
protease
productions. Some
pre-oxidize
soil
and
have a specific
phosphates
structure
while some
problem
other
which need a
enzymes are
proper
not
soil
affected
management
with the
practices
increasing
for a better
nitrogen
soil
fertilizer application.
um,
More often,
structure.
however, enzyme activities are increased when organic and inorganic nitrogen fertilizer or
Sandy soil. This soil have a problem of low water and nutrient retention. Retention. The practical method of improve structure of the soil, such as soils, is to
are
add
added
organic
together.
matter
Nitrogen
directly or
fertilization, especially in a mineral
through a
forms,
green
may have
manuring.
an indirect effect on the activity of the soil
The
enzyme.
addition
Why?
of the
Change in the
pond
soil properties
sediments
such as
can
the soil reaction,
clay soil or even a
dehydrogenous
compaction is
activity is
helpful
highly sensitive to
in
the
increasing
inhibitors effect
the
that are the
proportion of
association
small
with the
size
large
pores for
addition
the
of a mineral
enhancing
fertilizer.
uh,
Phosphorus fertilizer
water
under a
and
field
nutrient
condition
retention
has been
capacity.
shown to
Clay soil, these soils
decrease
have a
phosphate
restricted
activity in
drainage and
agriculture system. The
aeration
application of the
due to
lime
a poor
to soil most often
aggregations.
causes
Addition of a soil
causes a
matter through a helpful
significant
for the improving
increases in the
aggregation, but
pH,
the
and those
required amount
significantly
is
affected are
high.
the
Therefore, a
physical,
crop
chemical, and
rotation and use
biological
of
soil
phosphatic
properties such
fertilizers
as the
are
microbial
useful.
biomass and the
Raised or raised
diversity
bed
and therefore
cultivation
enzymes activity
can
increase the
reduce
protest and the
aeration
sulfates
stress
activity
problem
but decrease
to
the
some
phosphate activity.
extent. Um,
Organic amendments have a short term and the long term effect on the soil enzymes activity. The short term increase in the soil enzymes activity after the
Flood
application of the
condition in
organic
areas under
amendments can
prolonged
alternate to a greater
waterlogging, the
microbial
soil
biomass
aggregations are
due to
broken,
either
leaching
the
to
addition
flooded
of the
condition, a poor
microorganisms
drainage, and
and the
possible
enzymes and the
solution of a soil
amendments
problem.
are indirectly
Disparate
to the
condition, the
addition of
problem of dispersion is
available
found in a
organic
sodic soil.
and enzymes in the um,
Addition of a gypsum is a
available
combination with the
organic
green
substrate that
manuring
promotes
manures or
the
the
growth and
incorporation of crop reduce
indigenous
in a
microorganism
successful
and the
in the
long-term
improving soil
effect of the
structure and
organic
the
abatements of enzymes activity is
ameliorations of
properly
this
the combinated
soils. Low
effected of
stability
a
of the aggregation.
higher
Low stability of the aggregation in the soil of the
degree
airy
of
and
the
semi-arid
stabilizations of
region.
enzymes to humic substance and the increase
Due
in the
to a low
microbial
organic matter
biomass with
content,
the
stability of
increased
aggregates can
soil
be increased by
fixation
the
as well
addition of
as
the
the
organic matter.
inhibitors.
Mulching may also be
Effect
useful
of
as
heavy
it
metals and
provides
other
a
toxic
favorable condition
substance.
of the microbes to decompose organic materials. Influence of the texture and structure on the soil porosity and pore size
Uh,
distribution. The
Biological
porosity of
nitrogen
pore size distribution in the soil
imports
are affected by a
nitrogen
soil
imports
factors,
is the
mainly
agriculture
texture and
system is
structure.
provided through the industrial
The organic matter bulk
nitrogen
density
fix
and
in the
management
form
factor
of the
like a
chemical
tillage and
nitrogenous fertilizer.
cropping and
The
irrigation which
biological
affect the
nitrogen
soil structure
fixation
indirectly
bacteria
affect
is
the
a
total
natural
porosity and
or a
pore size
capable at
distribution.
reducing the
It has
atmospheric
already
nitrogen and to
been
ammonia
mentioned
with the action
that
and the
the porous
enzyme
textured
nitrogenous.
soil have a
The process is termed as the biological nitrogen
low
fixation,
total
also
porosity
called a
than the
BNF.
fine textured
It is
soil.
the esteemed that the biological nitrogen fixation contributes 140 ton
Although the size
gram of
of
nitrogen in
individual pores
agriculture and
is large
tertiary
in porous structured
ecosystem
soil, the
globally. Every
porosity in
year,
clay
whereas
soils is a
less
highly
than a
variable as
hundred
the soils
mm,
exhibit a
ton
swelling,
gram of
shrinkage, and
nitrogen is
aggregation, dispersion,
added
and
through
compaction
a
and
chemical
cracking upon
fertilizer.
wetting and
As a
drying.
thumb rule
The
of
fine textured soil have a large proportion
proportion
of
of
the
a chemically
microbial
and biological fixation nitrogen is a 50 ratio 50. The different nitrogen fixation organism and the symbiosis found in the agriculture and tertiary natural ecosystem and the important in a
proportion of
nitrogen
micropores.
economy.
Therefore,
A
this
biological
soil
nitrogen
retention
fixation in
higher amount of water
nature in the carried out by the various microorganisms in the agriculture as well as a natural system. Biological nitrogen fixation system in agriculture